In 2024, researchers mining massive databases of human and environmental metatranscriptomes found something hiding in the noise. They uncovered an entirely distinct layer of the subviral biosphere, a population of biological entities that had gone completely unnoticed. These newly defined entities are called RNA obelisks, and they are highly common. They are ubiquitous components of the human microbiome, with some studies detecting them in up to 50% of the human oral cavity sampled. Structurally, an obelisk mirrors the small circular RNA genome of a plant viroid. Yet while viroids are strictly non-coding, obelisks carry the capacity to encode their own novel proteins, a trait typically reserved for autonomous viruses. When you compare standard genetic material to an obelisk, the architectural difference is striking. Beside a sprawling, linear eukaryotic RNA transcript, the obelisk is highly compact. It is a covalently closed circular RNA, forming a rigid loop roughly 1,000 nucleotides long. By bridging the gap between simple viroids and complex viruses, obelisks represent a distinct class of autonomous replicons. Their physical structure and coding capacity challenge how we define the boundaries of life within the RNA world. To understand how an obelisk operates, we have to look at its baseline architecture. The architect, obelisk alpha, is built from a covalently closed circular RNA genome measuring exactly 1,164 nucleotides. But this circular genome doesn't remain an open ring. The sequence is highly self-complementary. The RNA folds inward, base pairing with itself to reach roughly 72. In dynamic environments like the human gut or oral cavity, cellular enzymes called ribonucleases actively hunt and degrade free-floating single-stranded RNA. The tight intramolecular base pairing physically limits exposed loops, protecting the genome from enzymatic cleavage. This data matrix shows how obelisk alpha aligns against known subviral agents. It shares the circularity and high base pairing of a plant viroid like PSTVD, but at 1,164 nucleotides, it is significantly larger. And unlike the hepatitis delta virus, which relies on a single ribozyme, the obelisk uses its extra sequence length to encode unique proteins. The obelisk's rod-like configuration is an active, engineered requirement that ensures the replicon's survival in a hostile cellular environment. Obelisks multiply without ever converting their genome into DNA. Instead, they rely on a mechanism known as symmetric rolling circle replication. Transcriptomic data reveals that this process involves the continuous synthesis of both sense and antisense strands. The host's hijacked machinery travels around the circular obelisk genome, spooling out long, multimeric chains of RNA in both polarities. To be functional, these continuous chains must be cut into individual genomes. This is achieved by the type 3 hammerhead ribozyme. These specialized RNA motifs are embedded directly within the spooled strands, and they act as molecular scissors, self-cleaving the multimeric chain into precise unit-length circular genomes. Once cut and circularized, the RNA must manufacture proteins without the standard 5' cap that host ribosomes require. To bypass this, the obelisk contains structures morphologically similar to internal ribosome entry sites, or IRES. These internal loops recruit the host's ribosomal machinery directly, initiating translation independent of normal regulatory pathways. Obelisks function as entirely distinct biochemical engines. By relying on embedded structural motifs, they overwrite the host cell's standard rules for both replication and translation. The defining feature of every obelisk discovered is the capacity to encode at least one protein from a novel family known as the oblins. The primary variant, oblin 1, is an essential component of their biological cycle. What makes oblin 1 unique is its structural consistency. Even when comparing obelisks from vastly different ecosystems, where their genetic sequences have heavily diverged, the three-dimensional structure of the resulting oblin 1 protein remains strictly conserved. This structural retention across diverse microbial lineages points to ancient evolutionary origins. Using predictive structure-based searches like DALI and FoldSeek, researchers have mapped the 3D architecture of oblin 1. It adopts a highly stable and well-defined core fold, maintaining its specific shape regardless of underlying sequence variations. A prominent feature of this core fold is a domain enriched with positively charged amino acid residues. This region is hypothesized to function as an RNA binding surface, magnetically docking onto the negatively charged phosphate backbone of the obelisk's own rigid RNA rod. By physically binding to the genome, Oblin-1 functions as a structural anchor. It stabilizes the RNA and masks it from sensors, demonstrating that the obelisk's nucleic acid and protein components are an integrated, codependent system. Because these entities were discovered computationally in complex metatranscriptomic mixtures, pinpointing the specific bacterial hosts they infect poses a significant challenge. The raw data provides the obelisk sequence, but not the address of the cell it inhabits. To locate the hosts, bioinformaticians track patterns of mimicry. RNA agents adapt their nucleotide composition, specifically their GC content and codon usage bias, to match their host. This data visualization demonstrates a clear match. The codon frequencies of an Oblin-1 gene map perfectly against the genetic profile of the bacterium Streptococcus sanguinis. This mimicry serves a direct mechanical purpose. By mirroring the host's preferred codon sequences, the obelisk ensures that the host's specific tRNAs are readily available, allowing for rapid and highly efficient translation of the Oblin proteins. Direct evidence of a hostile relationship comes from bacterial CRISPR systems, which maintain a genetic record of past invaders. Researchers have found obelisk-derived sequences permanently integrated into bacterial CRISPR spacer arrays, confirming that bacteria actively recognize and target obelisks as foreign threats. Obelisks are active participants in a biological conflict. Their presence triggers bacterial immune memory, forcing the obelisks to continuously mutate and escape recognition, locking them into a perpetual evolutionary arms race with the microbes they colonize. This conflict extends beyond simple bacterial immunity. The highly double-stranded character of the obelisk's rod-like structure makes it inherently vulnerable to recognition by RNA interference, or RNAi, machinery across various domains of life. In RNAi defenses, host enzymes like dicer and drosha scan for double-stranded RNA. The obelisk's base-paired rod is exactly what dicer is programmed to clamp down on. If masked by oblins, it evades cleavage. Alternatively, dicer severs it into 21 nucleotide small RNA fragments. If the obelisk is cleaved, those resulting small RNAs can be loaded into a host targeting system known as the RISC complex. Through a process called host-induced gene silencing, these fragments can guide the complex to accidentally or intentionally bind to and silence complementary host genes. Determining the exact nature of this interaction is the current frontier of this research. Cracking how obelisks navigate host RNA machinery will reveal whether they exist as silent passengers or if they actively modulate their hosts and drive pathology within the human microbiome. The search for obelisks has rapidly scaled beyond isolated datasets. By utilizing the Logan database to execute planetary scale alignments, researchers have already documented a 10-fold increase in known obelisk sequences compared to the initial petabase scale search. The resulting data reveals a global footprint. Obelisks have been identified surviving in biomes ranging from boiling hot springs in Yellowstone to the freezing depths beneath the Antarctic ice shelf, and they are remarkably durable within humans. Longitudinal studies track the same obelisk variants persisting within individual microbiomes for over 300 days, maintaining a stable presence despite the constant cellular turnover of the gut and oral cavity. This high environmental persistence suggests a future therapeutic application. Scientists have proposed treating obelisks as RNA plasmids. By leveraging their structural rigidity and cap-independent translation, researchers could potentially engineer these replicons into highly biostable vectors for targeted drug delivery. The discovery of the obelisk requires a re-evaluation of genetic taxonomy. They demonstrate that the ancient RNA world is a mechanically complex layer of biology actively replicating within us today.